Hello, this is Mistaken For. This is trial number 3 of this video. And we want to continue our discussion on DNA, and we're going to look at DNA replication. And the DNA replication, we know the basics, right? We did an example in class. If one strand is AAA, then the other strand needs to be TTT. So we understand the code of it, or at least we're beginning to. But what we need to go over are some of the mechanics. So go ahead and find this picture in your notes, and if you want, you can hit pause now and just go ahead and draw this. So if you had your notes, and if you've gone ahead and written all these things down, let's go ahead and real quickly go over that. So we have this job of the DNA helix, and we need to replicate it. And this job means that we've got to unzip the DNA. So we're going to unzip, we're going to use an enzyme called helicase. And the helicase is an enzyme that disrupts those hydrogen bonds. So those hydrogen bonds are there to keep the DNA helix together. So if it interferes with those hydrogen bonds, it's going to be able to separate. It's going to pull them apart and unzip it. Well, that's all well and good, but the DNA helix can simply come back together again once the helicase is passed. So in order to prevent that, we have single-stranded binding proteins, and they cover the single-stranded DNA, and it prevents it from coming back together. So it stabilizes that unzipping process. And then weirdly, we need we need an enzyme whose only job it is to kind of allow the tension in that coil to be released. So, for instance, if you imagine a strand of yarn, and yarn is made up of several smaller pieces. And if you imagine trying to separate that and pulling them apart, then what you get is a really super stressed area as you continue to pull it. And the same thing happens in DNA. As you uncoil it, as you unzip it, there's going to be supercoiling that happens at the two forks. So the topoisomerases, they break that backbone, and then they allow the DNA to kind of swivel, and it relieves the stress of that coil. So those are the enzymes that are involved in unzipping the DNA. On the right-hand side, what we want to look at is this primase, and this enzyme is solely there to build our RNA primer. And the RNA primer, just as the name implies, it's made of RNA. And that RNA is important because DNA, we don't have an enzyme that can build DNA from scratch. Like, okay, there's no DNA, let's build one, let's add to it. So, and we can go over why that is, but just for now, just go ahead and accept it. We need an RNA primer because DNA can be added to something, but we don't have an enzyme that builds it alone. So if we have an RNA primer, which can be done all by itself, then DNA can be added to it. Notice the arrow here. That means that we're going to be adding nucleotides to this side. That means this is the 3' end because we can only add to that. So the RNA primer is built from the primase enzyme, and it's about 5 to 10 nucleotides long. And then after that, we can add our DNA. So there is kind of our building of our replication fork. Now, there's going to be something interesting about our DNA strand. The DNA strand, remember, is anti-parallel, and that poses some problems for one side, and that's going to be this bottom side. You notice it just looks a little messier. Now, the top side is easy. The top side is the leading strand, and the leading strand, it leads it a little ahead of the lagging strand. And the leading strand is continuous. And that means it can always just add to the strand. Let me show you what I mean by that. I'm just going to go ahead and use my colors. The leading strand can just always add to this growing nucleotide chain. It will always just be able to add to the 3' end. But let's do a for instance. Let's go ahead and open the strand up to the next. So say in the next 10 seconds, let's look at this, what it would look like. So we've opened up the DNA strands. Now, if we do that, that means the leading strand can simply continue to build. So here I am building with my light blue. I'm just adding on, adding on, adding on, adding on, adding on, adding on. And no matter how much time, how much more I open it up, that's all it has to do. It can just add on. Well, that's all well and good, but let's take a look at what has to happen in the lagging strand. Now, the lagging strand, the 3' end, is on this side. Now, that's the key to understanding this. If the 3' end is going in the opposite direction, we're going to have a puzzle because, and follow along with the colors if you want, we're going to have to build my RNA primer, and notice this time I have to go in the opposite direction. Open, open, and this is our 3' end that I'm adding it to. So I can continue to do this until I hit the RNA primer that was there before. And now notice I've got a fragment of DNA. I don't have one nice long leading strand. If we did that again, then the same thing is going to happen. I'm going to open up my replication fork. I know this is going to look messy, but I think if you see me do it, you'll be able to get it better. The leading strand, again, is the easy one. We can just kind of keep adding and adding and adding. I should have done that in the opposite direction. That could be very confusing. I can't seem to get my eraser perfect. Oh, well, I'll redo that. I don't want to have to do this a 4th time. So there's my DNA strand, the open part, and the leading strand can build it this way onto the 3' end. And if we think about what's going to happen, we have to build our RNA primer again, and then we're going to have to add to our lagging strand again, and we're going to hit this RNA primer again. And so what ends up happening is you get these fragments. And the Japanese scientist his name was Okazaki, and he was the one that really kind of figured this out. He realized that there was always this one strand that was very long, and that was the continuous leading strand. And then there was one strand that always seemed to be chopped up, and that was the lagging strand. And we call these fragments Okazaki fragments. So there's our kind of our basic drawing. Let's go ahead and do this. This is our leading strand, and notice it's continuous. We can add on to it. There's our DNA polymerase happily adding on our nucleotides, and no matter how many times, because this is going to the left, we can always add to it. Now, the lagging strand is the frustrating one, so let's take a look at this in more detail. So here's our last picture that we need to go over. Now, again, here's our leading strand. It's continuous. There's our DNA polymerase. Let me zoom out so we can see that in totality. There we go. And so notice, and let's go ahead. I'm going to go ahead and draw it, but you can just follow along. The lagging strand down below, it's got to add another, here would be another primase. Like right here, we would be adding the primase and the primer. So it would be adding it right there. There's our primer. And here's where we're building another fragment. So let me get our light blue. We're adding our new DNA, the nucleotides, in this direction. And notice we're hitting another primer that was built. And so that would have been added on earlier. And it's hitting a primer that it opened up. And so we're getting these nice fragments of primer and then new DNA. Primer and new DNA. And if we want, we can do one more just to kind of prove it to ourselves. If this was opened up again, we'd have to do the same thing. We'd have to add our primase, our primer, and then the DNA polymerase is going to add DNA to this. And we'll go over some of the stats. This DNA polymerase, that can add things quite fast. In bacteria, it could be 500 nucleotides per second. In us, it's more like 50 nucleotides. Still pretty impressive. And then for the Okazaki fragments, they can be anywhere from 1 to 2,000 nucleotides in bacteria down to 200 nucleotides in eukaryotic cells in us. Now, do these remain? No. Look what happens towards the end. There is an enzyme that breaks down the RNA primer and converts it to DNA. And you're seeing that right there. It's removing that RNA and it's going to sew it up with the proper DNA nucleotide. And then there's another enzyme called ligase that can sew it up. And then you get a nice proper length of DNA. But this has to happen as it continues. It's constantly lagging behind. Now, that's a bit of a misnomer. It's so fast, it's keeping up with the leading. But we find that term convenient because it's always having to go in the opposite direction. So hope that helps. We're going to do an activity tomorrow that's going to really allow us to figure things out. Have a nice day.